Today we will discuss about MLKL-driven necroptosis will promote macrophages, extracellular traps, that is METs. Don't be confused by these METs as mesochemicals to epithelial transactions, although their meaning will be same as we go through the result sections. In the previous video we have discussed how the macrophages helps the tumor cells from phagocytosis, but just providing the phagocytosis resistance does not impart them metastasis. So they need to check how this metastasis are occurred in these MLKL driven tumor cells. So they from the results they made a hypothesis that there could be some intracellular changes that occurs that triggers these macrophages extracellular traps or some metastatic properties. So first they did the 3D invasion assay and migration assay, and in this case they find out that in the and these assays are done in the co-culture with the macrophages. In this case they find out that both for this overexpressed MLKL gene in the case of PANC1 and ASPC1, and for both invasion assays and migration assays they can see that when the MLKL are overexpressed there are more invasion and migrations, and these things can be reversed when they use the inhibitors for the MLKL gene. So this both the migrations and invasions in PANC1 and ASPC1 are reversed when they use the inhibitors for the MLKL gene, and these things are quantified on the right hand side, where we can see that for the PANC1 when the MLKLs are overexpressed they see that invasions are higher compared to the control case where there is no overexpression or inhibitors for the MLKL are used, and the results are similar for the ASPC1 in the case of invasion assays and for both the cases in the case of migration assays. And when they did the FN1 expression, which is the markers for the migrations in the cancer cells, they can see that when the MLKL are overexpressed they see that more expression for the FN1 compared to the control case. And they also check the markers. They also check the markers for this EMT, that is epithelial to mesochemical markers such as N-cadherin, and they can see that when the MLKL overexpressed they see the more N-cadherin markers, indicating that there is more mesochemical transition occurs. And when they use the inhibitors, these mesochemicals markers are reduced, and these same things they observe for ASPC, although the expressions of N-cadherin are lower is slightly visible. However, a difference can be seen that in the case of where MLKL overexpressed we can see some N-cadherin signals, but when the MLKLs when the inhibitors for the MLKL genes are used we see no signals. And when they co-culture when they did the in vivo experiments with the when they did the in vivo experiments on this anode SCID orthopedic models, not the in vivo, these are ex vivo experiment results. So in the ex vivo experiments result where they co-culture PANC1, what they have seen is that when the MLKL are overexpressed there are more chances of liver metastasis, and when they use these inhibitors for the MLKL the chances are low, and same things they have observed for the ASPC co-cultured cells. So these are the results. These are the results for the in vivo experiments where they transport this PANC1 pancreatic cancer cells, and in another they transport this ASPC1 pancreatic cancer cells. And this in vivo experiments has the more in vivo mouse models has the more more efficient immune system. That is why it is the non-SCID orthopedic models. And when they did the immunofluorescence of the co-cultured macrophages with these pancreatic cancer cells, what they have find out is that there is a formation of macrophages extracellular traps occurs around the macrophages that can be seen here, and some of the markers for the macrophages extracellular traps such as MMP12 and CH Th3 are found, and they are more predominant in the case where MLKL are overexpressed. So that can be seen here. Here the markers are more, and for the ASPC also markers are more when the MLKL are overexpressed. And these things are quantified in the case of on the right hand side, where we can see that when MLKL are overexpressed, the markers overexpressions are higher compared to the control case. In the control case, it around expressions are around this fluorescent intensity is around 11, whereas in the case of this MLKL overexpression intensities are around 70. And same things are for this ASPC MLKL overexpressions, where maximum intensities are around 37, whereas in the control case maximum intensities are around 11. And when they do the Western blot to check these markers for this macrophages extracellular traps, they found the same results, that is MMP9 in the compared to the solo culture. The co-cultures has the high expressions of this macrophages extracellular traps, that is MMP9, MMP12, MPO, CITH3, and these things are being reversed when they use these MET inhibitors, indicating that MET inhibitors might reduce this metastasis properties. And same things they have observed for this ASPC cell lines also, where these markers for the MET, these macrophages extracellular traps, are reduced when they use the inhibitors for this METI. And in the panel H, we can see that in the panel H, we can see that this markers these markers such as for the solo culture and for the co-culture, here the inhibitors for the MLKL are used, whereas in the panel G the inhibitors for the MET are used. And when they use the inhibitors for the MLKL, they see the similar results, that is the markers for the macrophages extracellular traps increase, indicating that when the tumor cells interact with the macrophages, then only these markers for the macrophages extracellular traps increase, and that somehow helps in the metastasis of the cells. So after that they check some of the markers for this EMT characteristics, such as cytox, orange staining, and they can see that when this MLKL are overexpressed, this is the more signals for this macrophages, cytox, orange staining, which is the markers for this EMT, this epithelial to mesenchymal characteristics. However, when they use these inhibitors for this MLKL, these overexpressions are reduced, and these things are quantified on the right hand side, where we can see that cytox orange are overexpressed when the MLKL are overexpressed. However, they are reduced when the inhibitors for the MLKL are used. And in the panel J, we did the in vivo experiments with these MLKL overexpressions, and the MLKL overexpressions with these inhibitors for the MLKL. And next they use this anti-lyse, and third they use the anti-macrophages, so that is clopsome. And what they have find out is that these markers for these macrophage extracellular traps, that is CITH3, is reduced when these MLKLs, either MLKL inhibitors are used, or this claustrophone or macrophages inhibitors are used, indicating that it's the interactions between MLKL tumor cells and the macrophages, tumor cells that have this high expression of the MLKL, and the macrophages will cause this metastatic favorable changes within the tumor cells. And next they check the EMT markers also, such as the encatherine and such as this encatherine, and they can see that when they use the MET inhibitors, the N-Catherine markers are reduced compared to the case where only MLKL are overexpressed and no MET inhibitors are used. And these results are uh, same for the PANC1 and ASPC1. And in the ASPC1, the markers, N-Catherine markers, are almost non-visible. However, the other markers, that is E-Catherine markers, we can see that there is an increase in the E-Catherine markers when the inhibitors for the MET used, whereas E-Catherines are low when the MLKL are overexpressed but no MET inhibitors used. And finally, when they did the mouse in vivo studies for where the immune systems is not so efficient, here they use the pancreatic cancer cells with a conditioned medium, pancreatic cancer cells with a conditioned medium that contains MET site MET. We can say that contains the MET upregulated cytokines, or that contains cells with MET containing conditioned medium. Yeah, METs. The METs, we can think of these as CITH3, MMP12, and MMP9, MPO. All these things they contains, and when they use this MLKL overexpressions, you can see that almost all the in all the cases they see the liver metastasis. However, when they use these MIT inhibitors, they see the reduction in the liver metastasis significantly. And same things they have observed when they use the when they do the in vivo experiment with ASPC pancreatic cancer cell lines. That is, when MLKL genes are overexpressed, they see that more or the liver metastases are high. However, when they use the MET inhibitors, and they see the reduction in the liver metastasis. So here the ASPCs are or the ASPCs are ASPC tumor cells are injected, which are co-cultured with this MET containing conditioned media. So the MET containing conditioned media will lead to the changes in the ASPC cells and also in the PANC1 cells in a way that will reprogram them towards these EMT characteristics, to acquire EMT characteristics. So I hope you understood this, how this MLK-driven necroptosis promotes this macrophages extracellular traps or metastasis in the pancreatic tumor cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.